Hello everyone, this is Farshid Salmi Jazi from Barstow Lab at Cornell University. Today I'm presenting the engineered electromicrobial energy storage system, which uses the electroactive microbe to convert renewable energy and atmospheric CO2 to biofuels. This conversion would occur through hydrogen oxidation via hydrogen molecules or direct electron transfer via bacterial nanowire to electroactive microbe. But first, I'm going to give you a short summary of why we are worrying about storing energy in form of from renewable energy sources. First is that to allow renewable electricity penetration, these cheap and clean energy sources to our electricity generation system, and also to ensure the supply of generation matches the electricity demand, which can be changed hourly, as you can see in this figure, or even seasonally or regionally in our system. And so it's all about the sustainability. We need to have a sustainable system. But the second question would be that how much of energy we need to store and at what cost? In our recent publication, we tabulated this question in regard of storing renewable energy chemically, such as in batteries or flywheels. Considering the main elements in the batteries or flywheels are lithium and zinc, simply you can see here that if we want to store 3 weeks of supply, current supply, we need about 14 and 11% of whole world fraction of lithium and zinc, which is impossible actually, in fact, because we need these essential elements for other purposes is that we need those. So we need to come up with another solution. And the solution that we can think about is that comparing all of these energy forms by energy by mass or energy by volume. As you can see here, that hydrocarbon molecules have high energy by mass, even energy by volume, and even the best lithium-ion batteries have just 2% of energy density of gasoline. Therefore, converting renewable energy, we think, to hydrocarbon fuels would be one of the best options to widespread this storage system for different purposes without worrying about lifetime or environmental issues or even storage capacity that we are facing in our chemical storage like batteries that we have. But what is the first example of having these hydrocarbon molecules in our system? In fact, biology gives us the first template for storing energy as hydrocarbon molecules. For example, biomass or microalgae. For example, the C4 biomass like corn or sugarcane can have the maximum 3.4% in real experiment, which is considered as a low efficiency. And the main barrier in having this low efficiency is self-shading effect that we have in our plants like C4 or C3 or even microalgae. The top cell preventing other cells from getting enough energy from solar to convert CO2 to biomass. Also, biomass conversion we have here has low efficiency, but also we are having some other challenges like land usage competition. As you can see in this photo, this is a microalgae farm that occupying a huge land area, and it will be in this issue that we can use this area for agriculture, wildlife, or energy, or having a solar farm, having a PV solar cell here. So we need to come up with an idea or an approach that can overcome all of these challenges and minimize this drawback as much as possible. So we are going to say how microbial electrosynthesis can address these challenges. First, in microbial electrosynthesis, we are not limited to any state or region. So we can use any form of energy source, not just solar like biomass. We can use solar, wind, wave, nuclear, or anything that we have around as a renewable source. And also we are powering microbe with nanowire or even with hydrogen as a molecule. So we are not having self-shading effect. And also another one is that in microbial electrosynthesis, we can use all seven CO2 fixation pathways that are in nature in cyanobacteria. As you can see, from 4-HB to wood young dog, all of them can be higher than C3, C4, and algae in our system. So it's showing that the efficiency is really much higher in using CO2 fixation pathway in microbial electrosynthesis. And another problem was that occupying land area, that vertical installation in our system, which means that underneath of the solar panel, it would give us that no land competition in our system. No, because the cells are powered directly via the bacterial nanowire in direct electron transfer or via the hydrogen gases. So we don't need to be exposed on the sunlight directly. We just need to convert this electron through cathode or other any nanowire or through other molecule like hydrogen bomb. And also we can convert this electricity and CO2 to biofuel or bioplastic, or we can convert them back to electricity. So we should not be worried about the lifetime concerns or even other land competition that here we have. The main barrier here is that how we are going to power our microbe. How the electricity from solar, from wind will go to the microbe to power them. Here I'm showing you the most two dominant mechanism here we have. One of them is a hydrogen oxidation, one of them is a direct electron uptake. Hydrogen oxidation happens when hydrogen molecule will go through the outer membrane and inner membrane of our microbe, like in Ralstonia, and membrane-bound hydrogen or soluble hydrogenase. These enzymes get this hydrogen, this electron out of this hydrogen and transfer it to the metabolism, and this metabolism makes this reducing agent like NADH and power agent like ATP for biosynthesis reaction. And in the redox, as you can see, hydrogen, the electron hydrogen has a high redox potential that can flow easily through NADH, which is our reducing agent, and then to a biofuel. But another one is direct electron pathway. That electron directly to the nanowire or from direct contact will come through the metabolism, then will go to make NADH or ATP. But the problem here we are facing is that the jump we have from low redox potential to high redox potential, which we don't know how it could happen in the nature, which is happening actually in metal reducing microbe like Shibonella. But we don't know which system is most efficient. That's why we, in our recent paper in constraint, the title is Constraint on the Efficiency of Electromicrobial Production, that we considered all of the internal and external parameters to calculate conversion of solar and CO2 to biofuel efficiency or even electricity to fuel efficiency. And surprisingly, as you can see, even for the highest one in hydrogen, almost the efficiency is the same in hydrogen and ET. Although we have that electron jump that I told you that we need some external energy for that jump here. So here is the question is that so if the ET and hydrogen are possible, why even we are bothered to face the hydrogen usage in our system? Because as we explained in our paper, that challenges we have in hydrogen is that its solubility, flammability, and scaling in our system. So we say that, okay, so it's better we move to ET, but the problem is that ET is not well known like hydrogen in our system. As you can see on the left, this is the outflow system in direct electron transfer, that we know how microbe can transfer electron to the cathode. We know which enzymes are engaged, but for inflow, we are not sure how electron will come through cathode to our metabolism, and how this uphill pathway or this jump from low redox potential to high redox potential happen. That's why the first thing for using this ET pathway or planting this pathway in other microorganisms is that to understanding how this electron flow is happening in our system. So first, what we did, we went through our knockout collection. In our knockout collection, we have about 40,000 mutants, transposon mutants, that they are non-essential mutants, that we know which genes, that we know some of genes and we don't know some of the unknown function genes that we have. Using this each mutant can help us to see which genes are essential in uptake pathway or in inflow or outflow pathway that we have. And by running simple assay with each mutant will help us to understand which genes are good candidate for our electron uptake mechanism. So what we did, we used AQDS assay. AQDS is a dye that it can be electrochemically reduced by getting 2 electrons and converted to AHDS. And also the color will change from white to yellow. So when the microbe will eat this AHTS and get its electron and give that electron to the external electron acceptor in media, the color should change to aqueous. As you can see in this figure for the wild type Shivanella that we have, that the yellow intensity will decrease to the white and then convert back to the yellow. It means that wild type Shivanella can uptake electron externally and give it to the from AHTS and give it to the external electron acceptor in media and turn it to white. And because we are running this assay under hydrogen environment, the hydrogen will diffuse to our media, and hydrogen will give the electron to aqueous and turn it back to AHTS. This is how it happened in wild type. It means that wild type can have this ability to get electron externally from AHTS to aqueous. So by this collection, we will go through all of the mutants that we have and disrupted each genes that we have in our system, and to see which genes will make this happen, or which genes will disrupt this mechanism. As you can see in this assay, for example, these genes, knocking these genes out or disrupting these genes out would not affect this process, and it means that these genes are not in electron uptake mechanism. But these genes, we can see that AHTS cannot convert it back to aqueous. It means that these genes is essential for our uptake mechanism, and it prevents microbe to uptaking electron. That's how this is our first check that to understand our candidates in our cell. Like for example, another example is these genes here, that it has stopped microbe to uptaking electron from AHTS and convert it back to aqueous. That's how we chose our candidate. After having this candidate, for a double check, we send these candidates. There are about 50 or 60 candidates, 50 to 60 candidates, and did the electrochemical test on each of them. What we did in our electrochemical test is that we measured anodic and cathodic current in our cell. For example, letter B is showing the wild type behavior in our system. It has an anodic current and cathodic current, and this one is the region that wild type can be. We measure that each mutant, how disrupting in those mutants can affect its current in cathodic current or anodic current that we have. And we just organize them by affecting those mutant on cathodic current. And from A2 to N or M or L, you can see that these interrupting in these genes would affect the cathodic current. So these genes are important for us. Some of these genes are some essential genes, and there are some known genes that we know why it's happening. For example, if, for example, Z be the electron acceptor gene, we know that, okay, it's clear that some of these genes disrupting some of these genes would affect this behavior, but some of them didn't. Like, for example, among some of them, they are not known genes, and they are unknown genes that we are going to pick those, run another assay, and examine those and understand which genes are essential in our system, which are not known till yet. That's how we pick our essential genes in our system and figure out which genes are important for us. That's how we can build this pathway in another microbe, or even optimizing this pathway for using in electromicrobial synthesis system by converting CO2 and electricity to biofuels or bioplastic. Thank you for your attention, and I will be always ready to answer your question.